Hi everyone, my name is Love and I'm here to do the part 2 of the practice midterm review for midterm this Friday. And to jump right in, I'm just gonna go down to number 13. We left off at 12, we left off at 13, so we'll start there. 13 is saying a new insect species is 2n equals 2 in its body cells. A female homozygous dominant for G and homozygous dominant for H is crossed with a male homozygous recessive for G and H. A diagram of the nucleus of the body cell in the F1 is shown on the right. One of the alleles is labeled G. What is the possible alleles could go in the box for the diagram to be correct? So basically we're trying to figure out what is that allele right there, right? Well, what do we know? The female was a homozygous dominant, right? So the possible allele it would have given out is probably just G and H, right? So if we do something like this, it would be a G and an H. So this is coming from the female. All right. And then the male has homozygous recessive for both. So the only possible combination of this would be little g, little h, right? So we'll have a little g and a little h, right? So this is the male, this is the female. When they combine into a zygote, we see this right here, right? And we know that since this chromosome we're looking at specifically has the allele big G, we know that the only possible other allele on that chromosome could be the big H in this case, right? So in this case, our answer should be B. So number 14 is asking us that most of the mass that is used by plants to make sugars is photosynthesis comes from the sun, bacteria, soil, water, or the air. Well, we know that mass in photosynthesis is in terms of producing glucose, which is essentially a combination of a bunch of carbons. Carbons comes from CO2 in the air. So our answer in this case would be E. All right, so 15 is a little interesting. So we're going to talk about muscular dystrophy and how, so it's an X-linked recessive condition, right? And a phenotypic normal woman whose father had MD marries a normal male. So let's kind of break this down. If a phenotypic normal woman, so since this is a recessive gene, you could be normal if you had, if you were heterozygous or if you were a carrier. And the only way to be a carrier in this case would be if you have a female, if you're a female, right? Because males are XY. So if males had the recessive condition, say this is little r, then there's no way they can carry it. They already have it, right? But females have 2X chromosomes, right? So one can be dominant and the other one could be recessive, right? And still show the normal phenotype. So in our case, we know that our phenotypic normal woman has this combination right here. And we're saying that she marries a normal male. So if he was a normal male, he would have a homozygous, not a homozygous, a dominant R on the X chromosome, right? So let's cross this, right? Because they're expecting to have a child. So cross them. This is XR, this is the Y. And we know that these two would be homozygous dominant. This one, this female right here would be a carrier. This male would be normal. However, this male would be would have it, right? Would be recessive for it. So in this case, would have the condition. So the question is, they find out through genetic testing that their child will have the disease. Okay, so automatically you can rule out anything that does not have the disease in this case, right? But we know that this combination will not give you the disease. We know that this combination will not give you the disease, and we know that this combination will not give you the disease. So the only way to get the disease is this combination, right? Being a male that has the recessive X condition, right? What is the probability that their unborn child is a female? Well, the only way to have it in this case, if you're a male, so there's no way you can be a female, right? So we know that our probability is most likely going to be 0%, right? All right, so number 16 is saying, suppose that when bacteriophage T2 infects E. coli, the entire virus particle enters the cell. All right, so this is a very key ingredient to the question. If true, which is another key ingredient, then the Hershey-Chase experiment would have absorbed phosphorus in blank and sulfur in blank after centrifugation. So you got to understand that, first of all, this is a hypothetical situation. We're talking about the entire virus particle entering the cell. We know in real life through the experiment that only DNA entered the cell, right? So when you're taking your test, you got to be aware of these questions, putting in hypothetical situations. Just because your notes say DNA was a transforming agent doesn't mean that that's the right answer to this question, all right? So this question is saying the entire virus particle entered the cell. Now, from our previous knowledge, right, we know that, so be backtrack. Proteins are labeled red, and the reason they're labeled red is because they're radiolabeled with sulfur, right? And DNA is radiolabeled blue, and the reason they were radiolabeled through phosphorus, right? Now, why? Well, you know that no amino acids carry the element phosphorus in them, right? So that way, if you radiolabel something with phosphorus, you know that only DNA in a bacteriophage will be radiolabeled with it. Same situation. You know that only amino acids contain sulfur, right? The amino acid cysteine. You know that DNA doesn't have any sulfur. DNA only has carbons, nitrogens, hydrogens, and phosphorus and oxygen, right? So if you look at the difference between two, that's why you radiolabel with a unique element that's associated with a protein or a DNA, okay? So going back, so this is our bacteria cell. And what's gonna happen is usually the bacteriophage or the virus comes in, right? And it latches on and it injects its DNA that it had here into the cell, right? However, this question is saying the entire virus particle enters the cell. So we'd see something different. We'd see something like this. So we'd see the entire bacteria right here, and the whole bacteriophage, the virus, would be inside. So not only will the radioactive sulfur be inside, the DNA, radiolabeled DNA, will also be inside the cell, right? Because this is the cell. Now, what do we do to test where these radiolabeled things are? We centrifuge them, right? So centrifuging is basically think of it as spinning stuff so fast that it gets organized by weight, right? So the heaviest stuff goes on the bottom and the lightest stuff goes on top, right? So the thing is, a key thing is you're not breaking anything. You're not breaking the cell wall or anything. So that means that this cell right here is very heavy because it has a lot of stuff in it, right? It has organelles, it has a lot of stuff. So if something went in the cell, it's gonna show up at the bottom, right? As a pellet, right? And if something did not go in the cell, then it's gonna show up in the liquid that we have here. And this liquid is referred to as supernatant, all right? Just some vocab. So in our case here, we're saying that the entire virus particle enters a cell. So where would we see both phosphorus and the sulfur? Well, they're both in the cell, right? So we'd probably see them in the pellet, right, down here. So in our case, we probably see red and blue down here, which would prove to us that the whole virus went in. So what answer choice defines that? Well, Hershey-Chase would have absorbed phosphorus in the pellet, right? Because it went in the cell, the DNA went in the cell, and would have absorbed sulfur in this pellet as well, because the whole entire protein went in the cell as well, right? So I would say answer D is your correct choice in this case. These are probably very common questions on an exam because it just requires understanding of the material and really test your knowledge on it. So yeah. All right, so number 17 is asking us which is correct about meiosis and or mitosis in humans, right? So either or both. So this one's saying both, right? So meiosis and mitosis occurs starting with cells that have 46 chromosomes in their nucleus. So humans are 2N, so their diploid number of chromosomes is 46, right? And we know that in mitosis, so I'm gonna label mit, we're going from 2N to 2N, right? And meiosis, we're going from 2N to N. So if you think about it in humans, either way, we're going to start with 46 chromosomes, right? So our answer in this case is A, but let's go through the other ones as well. Both reduce the number of chromosomes to 23. Remember a keyword both? No, only meiosis does that. A human fertilized egg undergoes meiosis first, then mitosis for development. That can't be true, right? Because you have a sperm, which is N, and you have the egg, which is N, and they conjoin to make a zygote that's 2N. Now, you started off as a little zygote, right? And you became such a big, like, baby in a sense even, and not all of you was made of sperm or egg, right? Because if it was, that means that your zygote only did meiosis, but you're not, right? You have skin cells, you got eyes, like eye cells, everything, right? And those come from mitosis, right? Because those are somatic cells that have your full genome. So there's no way mitosis would have occurred first. Meiosis would have occurred first. It's mitosis, right? So C is not right. D, human sperm undergoes meiosis in order to increase the number of gametes. So human sperm does not undergo meiosis, all right? Germline cells undergo meiosis. Human sperm is already haploid. It cannot undergo meiosis. Meiosis only is going from diploid to haploid, all right? So that's a very key thing to remember, all right? And then obviously that makes E not true either. So yeah, your answer to this is A. All right, so number 18 is asking us during the reactions of the Calvin cycle. So I just put up the Calvin cycle right here just for reference. Anyways, so A is saying light energy is captured by Rubisco to make a covalent bond. Light energy is not involved in the Calvin cycle. Calvin cycle is light independent, right? So don't worry about that. ATP hydrolysis powers the joining of CO2 to a 5-carbon sugar. Well, we know that ATP doesn't come into play in joining CO2 to the 5-carbon sugar. That's done by Rubisco, which doesn't require ATP. And then we got G3P molecules get rearranged to make RUBP molecules. So that's true, right? So we're taking RUBP here, right? And Rubisco over here attaches CO2 to the RUBP essentially. And then ATP is what hydrolyzes these, or ATP makes these into G3Ps, right? And we have a bunch of G3Ps over here, which essentially get regenerated into a 5-carbon sugar, RUBP, okay? So essentially, if you think about it, you're just rearranging, you're making G3Ps, you're making 6 G3Ps, and you're releasing 1, and then you're putting 5 of them back together to make 3 RUBPs, okay? Because RUBP is 5 carbons, G3P is only 3 carbons, right? So over here, you released your 3 carbons, and you're left with 15, which you can make into 3 5-carbon chains, which are RUBPs, okay? So C sounds right in this case. D, NADP plus becomes reduced in order to produce NADPH. This happens in the thylakoid membrane in light-dependent reactions by NAD plus reductase, so not in the Calvin cycle. And E, the main product that is produced by the cycle is glucose. False. A lot of people get this one confused. It's actually G3P, all right? Glucose can be produced by G3P because glucose is an intermediate of glycolysis, all right? And that can go backwards in glycolysis through a process called gluconeogenesis to make glucose, all right? So remember, G3P is actually the product in this case, all right? All right, so 19 is asking us a plant with the genotype given here assumes that the genes sort independently, all right? How many genetically distinct gametes can be made by this plant? Well, so we know that they'll sort independently, so to each is their own combination. They all have an equal chance of making that combination, right? So we know that AAA, either way, you're gonna end up with an A, all right? B or little b, well, that's 2 possibilities right there, right? You can have a big B and little b, so that can be combined with either a big C or a little c. So that's another 2 possibilities, right? And then D and D, you have no choice. You're stuck with a big D over here, and little e, you're stuck with a little e as well. All right. So the only things that can fluctuate are the B's and C's. So you have 2 + 2 combinations, 4, right? And I'll show you which ones. You can have a big B and a big C. You can have a big B and a little c. Or you can have a little b, big C, or a little b, little c, right? And so if you look at it, there's no other possible combinations of these alleles right here, right? So you have 4 possible alleles. I mean, 4 possible gametes, my bad. But yeah. All right, so here we're talking about photosynthesis. During the overall reactions of photosynthesis, what goes on? So let's go through each one. G3P gets oxidized in the thylakoid membrane, or spaces. G3P doesn't even come into play until Calvin cycle. So let's take that out, right? And plus, if anything, it's in the stroma. B, oxygen gets reduced in the thylakoid spaces. So oxygen, in fact, does not get reduced. Oxygen is made through the oxidization of water, right, in the thylakoid space, but not this case. G3P gets reduced in the chloroplast stroma. G3P is not reduced, in fact, in the chloroplast stroma. That wouldn't work in this case because G3P is, like, if it were to get reduced, it would probably get reduced in the cytoplasm of the cell, right? Because that's where you can reduce G3P and combine it to make glucose. If anything, if this sentence said carbon dioxide gets reduced into chloroplast stroma, that would make a lot more sense, right? So that wouldn't be the case. D, protons are pumped into the thylakoid space. Yeah, that makes sense, right? Where the electron transport chain allows protons to get pumped into the thylakoid space from the stroma. So yeah, active transport, by the way, quick review. E, water gets oxidized in the chloroplast stroma. Water, we only see it being in the thylakoid space in photosynthesis. So that's not the case. All right. So here we have 2 human autosomal genes. P and R show 20% recombination. A man inherited dominant P and R alleles from his father and recessive P and R alleles from his mother. Key to remember, we're talking about 20% recombination. So it's asking what proportion of gametes carry both dominant alleles. Well, let's go. So the father donated this, right? A P and R. So you can say the sperm cell carried this. So father gamete, all right? And the mother's gamete had recessive, right? So little p, little r. So this is probably the egg, right? So these combined and produced him. And that means his combination was heterozygous PP and heterozygous R and R, all right? So this is him. And what they're saying is what proportion of his gametes, okay? So let's look at what kind of gametes he has. And the way I like to do this when we're looking at like dihybrids is basically like the FOIL method in like math, like factoring, right? So P to big R, all right? Big P to big R. You can also do big P to little r, all right? Big P to little r. You also have little p to big R, so little p to big R, or you have little p to little r, okay? Little p to little r. So if you think about basically all his gametes, so in his sperm, he will have 4 types of combinations. So there's like different combinations that each sperm will carry, and one can have this, one can have this, one can have this, or the other one can have this. Now, which ones are the ones that are from his parents? Well, these 2, right? And which ones are recombinants? Recombinants refer to ones that aren't similar to his parents, right? You don't see a heterozygous in his parents at all. But these are both recombinants, all right? And what was the percent of recombination? Well, 20, okay? So this, only 20% would exist like this, right? You can say 20%, right? So if you're talking about each one, then you can say 10 for this and 10 for this. Now, we're not talking about that. We're talking about the dominant alleles. I mean, we're talking about dominant alleles specifically, right? And so what proportion of his gametes would show dominant alleles? Well, you'd look at the big P right here, right? Which is basically 40%, right? Because these carry 40%. So 40% of those, and basically 40% of the other ones. My bad. I misworded that. So yeah, as we're talking about, we have 40% carrying both dominant alleles, which is what this is asking. Okay, because if they said that there's 20% recombinants and these are the recombinants, that means the 80% have to be split up between these parental genes, right? There's a 50-50 chance. So each parental combination carries a 40% chance. And since we're talking about both dominant alleles, that's only present with big P and big R, which is a parental gene, non-recombinant, so 40%, okay? If you want to do it mathematically, you can just do this. You have 100%, 20% are recombinants, so you're left with 80%. All right. Now, they're saying that we're talking about both dominant alleles. Well, you know that the only one that has both dominant alleles is the father, right? And so you can divide this by 2, and you'll know that your father accounts for 40. The combination your father had accounts for 40% of his gametes, right? Sorry for the confusion there, but the answer is C. Okay, 22. Polymerase chain reaction, PCR, is performed to amplify this DNA template shown here. Primers have the sequence all Gs or RAs. What is the size of expected PCR product? Now, first thing is, you're probably wondering, why the hell do we have 2 primers? Well, let me tell you what PCR is in the first place. So imagine you have this entire DNA genome, but you just want to amplify one part of it. Let's say we just want to look at this segment to this segment, all right? So what do you know about any polymerase? Polymerases can only be they only work in the 3' direction, right? So if you're looking at the bottom strand, this is 3'. So that means your bottom strand will go from 5' to 3' this way, and will only be able to go this way, right? However, you're trying to amplify this gene. Like, amplifying means like you're trying to get specifically to that location, right? And you're trying to make millions of copies of it. Now you gotta remember that this polymerase won't stop here. It's gonna keep going down the whole entire strand, but we only wanted this spot right here, right? That's why we have to throw in another primer that starts at the top. And this one will take it from, so this is the 5' end, and now this is the 3' end, right? And this one takes it this way, right? So basically, you send primers on each end of what your sequence you want to amplify is, right? So in this case, we have G, so you know that that's going to bind to C, right? And you know that our other primer is A, so you know that that's going to bind to T, right? So you essentially can figure out here by these 2 primers that this region right here is what you're trying to amplify, right? Because this one will go this way, and the top one will go this way, and you'll essentially keep doing that until you get exactly that sequence over and over again, right? I know it's a little confusing, but that's just how it works. You need a forward primer and a reverse primer, so you can stop and meet in the and get the section you want. But our question is asking, what is the size of the expected PCR product in base pairs? So let's just count what we're amplifying, right? Since we know that one of our primers will attach right here, and one of our primers will attach right here, our sequence that we'll get at the end is only going to be this long, all right? From this primer to this primer. And so let's count how many base pairs that is. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. All right, so we have 30 base pairs, and that's our answer. All right. Sorry if I complicated things, but that's just how PCR works. I really recommend watching the Khan Academy video on it. It will simplify a lot more concepts. So yeah. All right. So this question is asking that there are similar number of males and females in the human population because. So essentially it's asking us why is there 50-50%, 50-50 chance. Okay. So let's break this down a little bit. Females carry an egg, right? Males make sperm, right? And the conjoining of these is what makes a male or a female, depending on what combination was conjoined. Now, females are XX, right? And males are XY. So each gamete will have, due to the law of segregation, right? You know that these each will segregate into a gamete, right? So meiosis will happen, and basically one will be X. So for the male, one will be X, and another one can be Y. And there's a 50% chance of this sperm being X or Y because you're just going to separate, right? And so the female doesn't really have that option, right? Because they're both X. We know that the egg will carry just one X. All eggs will carry one X chromosome, right? So there's only the male can determine the gender essentially, right? If the sperm is an X, then you're gonna get a female. If the sperm is a Y, then you're gonna get a male. And why do these separations occur? Well, it's through the law of segregation, all right? So yeah, and in the male germline, all right? So B. All right, so number 24, where would the complex shown on the right be found? Well, easily, the only place you guys have probably seen this is a photosystem. Everyone can recognize that, right? You just gotta remember where it is. This is probably a pretty simple question. It's in the thylakoid membrane, right? This question just requires like knowledge of the diagram of where photosynthesis, the light-dependent reactions occur. So say this is thylakoid. These photosystems are embedded in the thylakoid membrane, right? And it allows the electron transport chain to come because they allow the excitation of electrons when light is absorbed, right? So our answer is C in this case. All right, so in the discovery of the structure of DNA, the fact that the two strands are in a double helix was apparent from the experiments done by. Well, Watson and Crick sound really familiar when you're talking about double helix, but they didn't really identify the structure that it was a double helix, right? They kind of got all the recognition, but they weren't the real people that actually identified and saw it, right? What happened was Rosalind Franklin actually identified through X-ray diffraction experiments, right? And she saw the results of a double helix in DNA. And so it was actually a combination of both Watson and Crick and Rosalind Franklin that eventually figured everything out. But if we're talking about the person of the structure of DNA and proved that it was two-strand double helix, that would probably be Rosalind Franklin, right? That's it for our second half of the midterm. Check out the first half if you missed it. It was posted earlier. And good luck this Friday. And make sure to email all of the SIs before Friday if you have questions. We'll be more than happy to help. Thank you. Good luck.